Hello, IB Biology. Today we're going to be talking about muscles and movement. There are three basic categories of muscles: the skeletal, cardiac, and smooth. Today we're going to focus on just skeletal. We'll talk about cardiac when we get to the cardiovascular system, and we'll talk about smooth muscle when we get to the digestive system. So we want to focus on the structure and function of skeletal muscles. So first of all, if I look inside of a muscle kind of looks like a rope, where if I were able to cut it and look at it, it's one big thing full of small little things. So a rope is normally made up of small fibers that are all kind of twisted together. And this helps with the strength of it. And that same thing is true for skeletal muscle. So on the outside, you've got a big muscle, but it's made up of these little bunches called fascicles. And inside each fascicle is a muscle fiber. And those muscle fibers are made up of myofibrils, and myofibrils are made up of sarcomeres. So we're going to look at a picture in just a second that kind of shows that kind of organization. Around the outside of a muscle fiber, which each individual muscle cell is called a muscle fiber. So muscle fiber, muscle cell, same difference, is covered by something called the sarcolemma. This is kind of the specialized muscle plasma membrane that has some unique functions, which we'll talk about at a different video. We also have something called the sarcoplasmic reticulum, which is really important for storing and releasing calcium. And when we talk about how a muscle contraction actually pulls together, we'll talk about that calcium, that sarcoplasmic reticulum. Okay. You might remember from your junior year in IB Bio that you talked about some exceptions to the cell theory, and muscle fibers are one of those exceptions. They are an exception because they're very long and they're multinucleated. So if you think about the rectus femoris, which goes the whole length of my femur, that muscle cell is that entire length. So it's really long, but it's also really skinny. So here on the PowerPoint, we see a picture of what a muscle looks like. So each one of these little circular things up here is called a fascicle. So this is a fascicle. And that fascicle is full of muscle fibers. So each one of these up here is a muscle fiber. If we zoom in on the fascicle, we'll see right here is the muscle fiber. And that muscle fiber is made up of little things called myofibrils. And so it's kind of like a repeating structure of inside of a muscle are fascicles. Inside of a fascicle is a muscle fiber. Inside of a muscle fiber is a myofibril. So you just have these kind of nested inside of them. And this is what makes muscles so strong. Inside of that myofibril, we have little things called sarcomeres. A sarcomere runs from one Z line, which is all the way here on the edge, to another Z line, which is all the way over here on this other side. We have little thin fibers we call actins. The actin is the thin fibers that are coming out from the Z line. And in the middle, we have these thick fibers called myosin. The myosin has these little heads on them right here. And those myosin heads are going to grab the actin and pull. So when we have a contraction, it's going to grab it and it's going to pull it together. So this H zone, this opening between that during the contraction, is going to disappear. So the myosin grabs the actin, it pulls it together, and then once we're done with the muscle contraction, it's going to let go. And so you'll see down here at the myofibril these little openings with the myosin that's a little bit thicker. When we pull those together, those thick places are going to get closer and closer together. Okay, so we're going to talk more about how this actually happens, but we want to make sure we have the sarcomere structure. We have that drawn because that's a common thing that they'll ask us on our IB biology test is can you draw a sarcomere? Can you label a sarcomere? So you've got to be prepared to do either of those things. So I need to be able to label the actin, the myosin heads, the Z line on the side, this H zone in between the two actins. Okay. So if I can label those things, then I should be pretty good to go. All right. So hopefully this is a good introduction to the structure of the muscle and of the sarcomere. Okay. We'll have a Zoom on Wednesday to talk about how this happens, and we'll also record a video at that time that tells me how contractions occur.